Good morning. CSIR life science memory based questions we were talking about. And one of the question was distance between two genes is 30 centimorgan. What it means is options were 30 million base pair apart, 30% chance of recombination, 30 base pair apart, or 30% linkage group, isn't it? So, talking about the question, in level of organization in biology, these is a increasing complexity hierarchy, isn't it? So, smallest is the atom, then molecule, macromolecule, cells, tissue, organ system, organism, and so on in the increasing levels of sizes and comparatively increasing level of levels, isn't it? So you must have seen the scales, and you must have known the what a centimeter is, what a millimeter is. In the scale, these line 1 2 3 4 5 6 7 that you see is what it is. It is a centimeter, isn't it? So on that scale, see the cell, a unicellular structure, that is either it can be a plant cell or an animal cell. It resides in the micrometer scale. So what is micrometer? Let's just break it upon a little bit. Okay, so 1 meter, 1 meter is equal to 100 centimeter. What is 100 centimeter? You have seen the scale, and what I just told you, 1 2 3 4, what that is written, that is centimeter, isn't it? So 1 centimeter, again 1 centimeter is further divided into how many parts? 10 parts, isn't it? So the smaller unit that you see in the scale is known as millimeter, isn't it? So 1 centimeter is equal to 10 millimeter. And again, 1 millimeter, when you divide it 1000 times, isn't it? You can see here the smallest scale is 1 millimeter. Then 1 millimeter, when you divide it 1000 times, then the scale that you reach is 1 micrometer, isn't it? So we are talking at the micrometer level, isn't it? So you can see here at this level, 10 micrometer around the normal animal cell, may be a plant cell. At that stage, at that scale, there that the animal cell or plant cell resides. And looking upon forward, it is, you can see here nanometer. Again, micrometer is 10 to the power minus 3, and furthermore 10 to the power minus 9 is uh, DNA, isn't it? So we are talking at the nanometer scale. So now you know at what scale we are talking. Okay, so you know about what the cell is, animal cell is. And in animal cell, inside the cell there are various type of cell organelles, Golgi complex, centrioles, nucleus, mitochondria. So inside the nucleus the DNA is present, isn't it? Inside the nucleus the chromosome is present, and in the chromosome is made up of DNA, isn't it? And a part of the DNA is known as gene. So let's say you have how many type how many what the chromosome, how many number of chromosomes do humans have? They have 23 pairs of chromosome, isn't it? So in male, 22 pairs plus XY, and in females 22 plus XX, isn't it? So 23 pairs in on altogether. So what are these chromosomes? These are just you can compare it with a type of books, isn't it? So one chromosome can be one book, and you have 23 pairs of chromosome, that is 23 pairs of books, you can say, so as to understand the, and what are in this in that chromosomes. In that chromosome contains the information of life, as the book contains information about your syllabus, isn't it? So let's jump upon the question, distance between two genes. Now you know what gene is. Gene is the part of the DNA, a small part of DNA in which the information about your whole morphology is characterized. Say some people have some people have white hair, some people have brown hair, some people have golden hair, isn't it? So some people have black eyes, some people have brown eyes. So these information are encoded in your DNA, isn't it? And in the DNA, the small part in which the information is encoded is known as gene. So that is the question. Distance between two genes is 30 centimorgan. It means 30 million base pair apart. The genes are 30 million base pair apart, and the 30% chance of recombination, 30 base pair apart, or 30% linkage. So base pair apart is not a correct interpretation, correct option, because you can see in this DNA structure, in lower figure that you see, there are lines in this. Is a ladder like structure, and the steps that are given in the ladder is what is base pairs, isn't it? Adenine, cytosine, thymine, and guanine. So these distance between these base pair, that is distance between these ladder steps, is known as base pairs, isn't it? So base pair means a base pairs with T and C base pairs with G. So these distances are known as base pair, and it is not known as centimorgan. Okay, so the option number B is, option number A is wrong, and option number C is also wrong. Okay, so coming upon option number B and D, isn't it? So linkage group, it is given. Linkage group means the tendency of gene to be inherited together. So you have seen in what do we say, in meiosis you have read, when you have seen, when a, let's say there are two parents, isn't it? Two parents, one, there are here it has been shown that one of the chromosome is from the parental maternal side, and one of the chromosome is from paternal side. Okay, father side one chromosome is, and from one of the chromosome is from mother side, isn't it? So you can see here, you can see in light blue color chromosome there is written capital A, capital B, isn't it? And in maternal side, let's say for example the small a and small b is written, isn't it? So after the meiosis, and after this chromosome undergo meiosis, you can see the letters have been exchanged, isn't it? Small b comes to the maternal side, and capital small a goes to the other side, isn't it? So this exchange of the property is known as, what it is known as, recombination, isn't it? And this recombination takes place in meiosis. Okay, so this just one concept, you remember this. And let's jump back to the linkage, isn't it? So linkage is the tendency of gene located close together on the chromosome to be inherited together during meiosis. So when two genes are located nearby each other, then the tendency of them going to or inherited into the future generation, inherited into the child progeny, okay, babies. So this tendency is known as linkage. So you just remember, there are 23 chromosomes and there are 23 linkage group. Okay, so when two genes are present in one chromosome, it is known as linked genes. Okay, and it has a percentage of 100% percentage 100% chance of being recombination with the other. The further it is, the more it has linked chances of recombination. So you will get clear when you go into deep. Just you remember that linkage group means there are as many the number of chromosome are there present in the organism that is the number of linked group. Okay, so for example, in humans 23 pairs of chromosome are present, so the linkage group is 23. Okay, and the percentage of the recombination chances is known as it is what do we say, it is uh, interpreted in, it is called upon by recombination property is called upon have a unit, is it is known as centimorgan. Okay, so what you have learned, you have learned that genetic linkage, you have learned genetic linkage is the tendency of genes to be inherited together. When does this linkage is related, when this linkage is related to the recombination, recombination it is related. Okay, so what is recombination? Recombination is the process in during meiosis when DNA strands are broken and repaired, producing new combination of alleles. Why recombination is important? For variation in the children, isn't it? And due to recombination, more strong and powerful genes are brought forward in the future generation. Okay, so genetic recombination is measured in centimorgan. You know, this is the explanatory image of where the recombination takes place and synaptonemal complex are formed and the crossing over takes place, isn't it? You have read this in class 12, and homologous recombination I just explained. So 30 linkage group is an incorrect option, and the chances of recombination is measured in 30 centimorgan. Okay, so option B is correct. Thank you and stay tuned for more.